Ladies and gentlemen, the opening program will commence shortly. And for the smooth running of this program, we ask everyone to please be seated and make yourself comfortable. And please kindly switch off or put your mobile phones on silent mode. Your cooperation is appreciated. Thank you. Ladies and gentlemen, good afternoon and welcome. His Excellency Professor Naoki Tani, Senior Researcher of the Forestry Division of JIRCAS. His Excellency Professor Yossi Kosumura from Tsukuba University. His Excellency Dr. Toshiaki Kondo, Senior Researcher of the Biological Resources and Post-Harvest of JIRCAS. Honorable speakers, distinguished guests, expert participants, ladies and gentlemen, and we have participants attending online as well. Good afternoon to you all. Welcome to Faculty of Forestry, Universitas Gadjah Mada, and welcome to Satreps Conference entitled Strengthening Tropical Forest Resilience based on Management and Utilization of Genetic Resources Capable of Climate Change Adaptation. We have now come to the most important part of today's program. We will have a plenary discussion with several experts. On the stage, all the experts have been seated to provide information and explanation according to their respective fields. To begin the discussion, we are pleased Dr. Widiatno to chair this plenary discussion for the next 75 minutes. Dr. Widiatno, please.
Excellencies, Professor Dr. Nauki Tani, Senior Researcher in the JIRCAS, Excellencies, Professor Yossi Kosumura, Professor Genetic from the Tsukuba University, and then Dr. Sosiaki Kondo from JIRCAS, and then Shinji Kakuta Sumitomo from the Sumitomo Forestry Crop, and Professor Muhammad Naim. Today I would like to say to give the opening about the research project collaboration between Faculty of the Forestry Gajah Mada University and then the JIRCAS already get the grant from the Satreps project, where the title is the Strengthening Tropical Forests and the Resilient based on the Management, Utilization, and Genetic Resource Capable on Climate Change Adaptations. Actually, why we choose this topic, because we know that our tropical rainforest is one of the largest tropical rainforests in the world, and then we have the many biodiversity, and then even though the number of the tropical rainforest in Indonesia less than 10% in the world, but we can contribute more than 50% biodiversity in the world. Just why how to keep and the tropical rainforest in Indonesia is very important, and then to sustainable our life in the future. And how we know that the importance of the ecosystem services. Some reviews say that the ecosystem, the biodiversity can contribute for many advantage. For example, when we have the high biodiversity, we can contribute for the ecosystem process, like the nutritional cycling and the movement of the pollen and the seed dispersal, and then production of the plant animal, and also how they can provide some many service like the fresh water, genetic resource, food, and other function. That's why the ecosystem and the genetic diversity and also biodiversity is very important for sustainable forest management especially for Indonesia. However, the forest degradation in Indonesia is very high. We know that for example, in the 1990 until 2010, we can see the decreasing number of the forest in Indonesia, and then if the forest is decreased, I think we have the many disadvantages like many secondary forests, fragmentation of the remaining forest, and create the small and isolated population, and maybe we expose the high the local extinction risk. For example, we know how the tiger in the Sumatra become lost, and then some species in Indonesia also become endangered species. So I think we need to keep the forest, especially tropical forest, to exist in the future. And then in this project, actually we have the many activity, especially how we can select the development trees capable to climate change adaptation using genomic selection. This is the new method to select the individual have the high growth and also how they can adapt with the climate change. And then after we select the good individual, the good clone for the forest plantation, we will do the mass propagation by the vegetative propagation. Hopefully, select this clone or the species
recording in progress.
The various environment, and also will adapt the climate change in the future. And then we also try to measure about the impact of the forest rehabilitation, not only in the forest protection, but also how the impact for the carbon sequestration and also the social economic. Especially in Java, we know that more than 40% population in Indonesia live in Java Island. That's why the forests have the important value to support their life, especially the food available of the food. So some project already done by the Prof. Naim and also Dr. Budiadi with the security food, with calling the integrated forestry and farming system, how we combine thick and also some of the agriculture species planted under the thick can contribute for sustainable human life. And then this is for instance how the silviculture system Dipterocarp in the tropical rainforest mainly dominated by the Dipterocarp, and then we already manage in the selective cutting but not sustainable. We can see the number of the forest concession become decreased, the number of the forest area become decreased. That's why new method I think is very important, especially how we can enrich the species with the native species in tropical forest is very important. Because we know that some impact of the selective logging, especially will reduce more than 50% the big tree, and then loss above biomass, and also declining species richness, and also genetic diversity. And another project, I think we already tried to find the suitable in the adaptation, and then we choose the good clone, I think, with the genomic selection. This is the new method for propagation and the large scale plantation, especially in the Java island. Yeah, it's also in the thick plantation. This is an example of the Dipterocarp forest in Indonesia. I think this is an introduction for me. Hopefully, we can enjoy from this seminar, and then another speaker will share with us how this project can be done, and then can contribute for the sustainable forest in Indonesia. Thank you.
Thank you, Dr. Widiatno. Uh, the next uh, the next session, can we invited Professor Dr. Muhammad Naim to explain about the current situation of forest and forestry in Indonesia.
I feel be in Scuba University because Professor Mura is my senior, and Prostani is my junior, so I feel to be in the Suba University. Yeah. um, okay, I will share the material. This is the topic of our project. This is the topic of our Satrep project. And of course, initiation of this project is actually already available, even of course not so completed, but some activity have been done, and team of the startup project then be make more complete the initial activity that's already happened, already done. This afternoon, I would like to, Pak Vidi asked me to present the current station of forest and forestry in Indonesia. Yeah, as I mentioned in the seminar before, that the condition of our forest in Indonesia is mostly dominated by species of the tetrapod because we have very large area of tropical rainforest. But we know that the productivity of species in the tropical rainforest is very, very low, only around 0.2 to 0.6. So even the rotation period is 30 years, but if the productivity is very low, so there is no meaning, because during one cycle, maybe the additional increment of diameter is very, very low also. So this picture in the below slide mentioned about the condition of our tropical rainforest. And then the number of tropical rainforest size, is about 120 million hectares. This figure is sometimes not fixed, sometimes increase, sometimes decrease. That's depend on. But the most important of this figure is we still have production forests. Yeah, at that time still to 23 million hectares, but now it's decreasing. It's about maybe around 18 million hectares. But this forest is very important because compared to the other Southeast Asian country, maybe Indonesia, the tropical forest in Indonesia is still number one, I think. In the world, the number of tropical, the Indonesian tropical rainforest is number 3 after Congo. Yeah, the first is Brazil, and then Congo, and then the third is
Recording
Indonesia.
in progress.
But in terms of genetic or biological diversity, it's recorded that Indonesian uh, biodiversity in tropical rainforest is to be number one. Since 1992, actually, our tropical rainforest is declining because of some reason, like logging, like forest fire, and also absent in planting forest activity, shifting cultivation, and other. So since 1992, from at the time 3.4 million hectares, becoming only 26.1 million hectares. Then from time to time, from year to year, the number of our forest concession or area in forest concession is decreasing, and right now this area is only about 18 million hectare, and also productivity is very low. Yeah, 7.1 million cubic meter. Ladies and gentlemen, because of that, and because of the productivity of our tropical rainforest species is very low, so we try to find find a solution how to increase our forest productivity. That is by some conventional method like species trial, and then provenance trial of different species have been done, and the result is actually very good and surprising. Why? Because from the species trial done in 5 concession holder model, we found that among 30 species of Soria, species of the Kalpase, we found at least 5 species that has very fast very fast growth, namely Soria leprosula, Soria parvifolia, Soria jorensis, Soria platycladus, and also Soria macrophylla. Those have around more than 2.5, and also some of them is also more than 3 centimeter diameter per year. So just only 10 years period, we will have 30 centimeter diameter increase. So that's why we use this approach to increase our forest productivity. If the increasing our forest productivity is not to be done, that means like in this figure, you can see that Meranti with the diameter more than 50 centimeter is only 2 trees found in 1 hectare. Yeah. So that followed by the diameter between 40 to 50, and 30 to 40, 20 to 30, and others. But mostly the number of trees per hectare is not increased. Just about less than 6 trees per hectare. The most rich the most plenty of species in 1 hectare is dominated by Meranti with the diameter 10 to 20. So this phenomena is very serious, give a back impact on maintaining forest productivity. That is why we try to improve the production of our tropical rainforest. So number of the area and also the number of concession holder also can be seen in this figure. That means right now the production per hectare of the tropical rainforest is around 30 cubic meter per hectare. But in the first time when we invite the company to work with the tropical rainforest, in the first time is the productivity is 60 until 80 cubic meter per hectare. So now is if company can produce 30 or 20, 25 or 30 cubic meter per hectare is still very lucky. This also happened in timber estate company. That's only around 80 until 100 cubic meter per hectare per cutting rotation. Usually for timber for timber estate company is only the cutting cycle will be 6 until 8 years. So in my opinion, both for tropical rainforest or plantation, in timber estate company, the production of species there it should be improved. One of our method to increase our productivity is using intensive silviculture technique. So this means the techniques is using the combination between three main silviculture elements, namely tree improvement or species improvement, and then environmental manipulation using fertilization, distance, using optimal using as optimal distance and other. And then we also use pest and disease management approach. And usually this activity is done especially in nursery. So we call it with the accelerated optimal growth activity. And if in the nursery we use accelerated optimal growth approach, that means that the quality of seedling that we will plant in the field is quite good. And then we continue to prepare good preparation for planting. Standard of plantation pool, standard of fertilizer, and then also planting time, the proper planting time. So the result, you can see that the Surya macrophylla or Tengkawang, even just 8 years old, the diameter is very big. This increasing productivity is not only happen in the ceiling using line planting pattern, but also ceiling with the gap planting system. So this the key of our method to increase our forest productivity. This is the condition of log of area actually. If we, we you know, this condition of our log of area is becoming bad and going worse if we we don't you know, we didn't use anything. But sometimes this condition of forest will continue to be palm oil, for example, and also for the forest area that can open access, so people can go there every time by bring the seed of many species that they want. So the forest condition is becoming loose and cannot be even company is not so interested with this condition of tropical forest. So that's why we use the line planting with usually between line planting is the distance is about 20 meter, but yeah, I mean tree within the line is 2.5 meter, but one line to the other line is about 20 meters. So we already set up the roadmap of tropical forest activity. Even we start from 1994 until now, still not complete yet actually. But we believe that finally we can produce the tree with the big diameter. So 20 years old of Meranti, of Soria leprosula, for example, the diameter is becoming 40 or 60, 40 or 50 centimeter diameter. This, plantation, big plantation in one company in PT Sari Kusuma, we proposed that in 2034 we will try to ask to the government, to the Ministry of Environment and Forestry, to give permission if we want to do the first cutting in that area. So we hope by experiment, this experiment maybe will stimulate the other company to follow the PT SPK PT SPK company. This is the performance of a line planting system. It's very nice, very good, and of course this method can improve and increase the productivity. And we estimate that 1 hectare may be 150 cubic meter compared to only 25 cubic meter per rotation. So about 6 by compared with the conventional management. Besides the tropical rainforest, we also concern on teak plantation, because teak is like in our what we call our in front of our faculty. I means the location is not so far. Yeah, if we want to concern on Meranti, on the capacity, that means we have to go to Kalimantan Island. If we want to concern on Merbau, we have to go to Papua. So TIC is really it's very close to us. So that's why we can make a concentration to do any kind of you know improvement of TIC. So until right now we have the program not only increasing our wood production of wood, but also we try to have what we call it integrated forestry farming system, combined between TIC and crop in one area. So this is the result that is very surprised actually, because this is the plantation in Ngawi district in compartment 7, just 20 years old, but the diameter is close to 50 centimeter. So, we predict that one tree is produce 2.5 cubic meter, or yeah, maybe 2 until 2.5 cubic meter. So if 1 hectare is we plant 200 cubic meter, so the production will be 400 until 500 cubic meter. That value is close to the radiata pine in New Zealand actually. So we believe that maybe using Meranti and using teak, our forest production will be increased. Yeah, if we try to connect with the Satreps project, maybe by using the genomic selection, our plantation in the future, of course, will increase spectacularly. Yeah, because the genetic material can already selected, and we will have, of course, better and genetic material. This is the the impact of social economic in our forest plantation in the future. We hope that by Satreps project, we will more increase our productivity and make benefit to the people. Yeah. As I mentioned before, that we concern on tropical rainforest, Dr. Karpase. We also concern on teak, but of course we also have to concern on pines, because pine is Mercury especially is native to Indonesia, and it can produce even wood for construction and also resin for for any kind of non-wood forest product. So this is the conventional breeding activity that we done since 1976 actually, and until now we still continue concern on gum rosin production. And yeah, we also emphasize on productivity, because we believe that productivity is very important. So that's why we also try to select the pine tree that produce more than 50 gram rosin per 3 days what we call reduce the tapping period, yeah. And if the rosin production is more than 50 gram, that we select those to be clusters of pines. We call it with the bocor getah means that pines can produce rosin, much rosin, produce much rosin. I think that's just information about some initial activity that and then by Professor Tani and team of Satreps try to write a proposal, and now the project have been accepted by JICA with the 5-year funding. I think that's Pak Widi, my presentation, and I'm sorry because yeah, it's not so cannot be a good arrangement, but I'm sorry for that. Thank you very much.
Thank you. Thank you, Professor Muhammad Naim. The next is Professor Dr. Naoki Tani from JIRCAS. Will explain about the tropical tree breeding using genomic selection and perspective of adaptation for climate change. The time is yours.
Thank you very much for your introduction. My name is Naoki Tani from JIRCAS. Thank you for your attention. So today I want to share with you the project about Satreps, and in this project, project, I'm leading the one sub project, which is genomic selection for the tropical host trees. So now the genomic selection, before the genomic selection, we have to make a model to speculate phenotype from genotypes. This is the genomic prediction model. So now this is next generation sequencer. This sequencer can create a lot of data. For example, this Illumina HiSec10 model. This can produce 1.6 to 1.8 terabase per run. This quite powerful tool can open the door to read a lot of sequence from a lot of individuals. So in the genomic selection, we make this is a training population, and then from the tropical training population, we read the sequence. This is the genotyping. And also, in the training population, we measure the phenotype. For example, in the tree, the growth is very important. We measure the DBH. This is the diameter of breast height, and also the height. And also, we can measure a lot of phenotype. And then we make a modeling. This modeling can predict the phenotype from genotype. But we have to verify this model. So we separate test population from the training population. And also, we conduct genotyping and phenotyping. But phenotyping, we use this model from genotyping. And then we can verify this model is correct or not. This is a genomic prediction model. So finally, if we can make this model, we can use this model for the selection. So for example, this is a lot of seedling. And then seedling, we collect the genotyping, but we don't know phenotype in the future. Because tree grow, it takes a lot of time to grow. But if we have this model, we can speculate this phenotype using this genomic prediction model. Then we can select this seedling. So this is genomic selection. So we can accelerate the tree breeding, because normally we have to wait a lot of time until this seedling growing up. But we don't need to wait in the genomic selection. So we just skip this one. And then I want to share with you about the current result of the genomic prediction modeling. So now currently we have some data from the, now Dr. Naim introduced you about this one company in Salvimixma in central Kalimantan. So the Dr. Naim established progeny trial, some species of dipterocalps. So now we targeted 3 species, Shorea platypulados, Shorea leprosra, Shorea macrophylla. So Shorea platypulados is studied by the Dr. Savitri, UGM, and Shorea macrophylla studied by the, my student in the University of Tsukuba. So then we collected a lot of the genotype using this RADOSEC technology, but this is a very detailed, so I can skip this one. Then this result from the Shorea leprosra. This is a progeny trial of Shorea leprosra. We use 254 individuals. So we divide into 2. One is a training population. We use 204 individuals. Oh, sorry. So then we use the 50 individuals for the training test population. Then this one is there are many types of the prediction modeling, but in this case we use a linear prediction model. So we call this is a Bayes ridge regression model. So if we use a linear model, the speculation is very difficult. This is data of the DBH. We cannot speculate. This one is we use the old marker. This is a 9,205 marker. And this one is we select this marker. This is a very informative marker. And then we select 384 marker. Still, we cannot get good model because this one is the test prediction ability is very low. However, second step, we use this neural network. This is a convolutional neural network technology. This one is now you are very familiar about face identification. Normally, the convolutional neural network used for the face identification. Your smartphone also has the CNN, convolutional neural network. Then we use this neural network to speculate the prediction, to speculate a phenotype. If we use this old marker, 9,205, still the prediction ability low. But we select the informative marker, 348, prediction ability is much increased. But still but low, but in this prediction ability, we can use for the selection. So then, this Satreps project, one of the important topic is we try to identify the physiological trait which is related to the climate change. So that's why now we are interested in some leaf morphology and the stem morphology. This is, for example, this is a wilted point, because the wilted point is very close to the stress of the drought. That's why we want to measure some leaf morphology, and also this is a stem. This wood xylem conductivity is very important for the water stress as well. That's why we want to measure this type of the morphology, and then we want to apply this morphology into the genomic prediction model. This is one of our target for the climate Satreps project. So finally, this is a workflow of the genomic selection. This is a target species, and then we set training population and testing population. Then we measure the phenotype, and also we measure the genotype using the next generation sequencer. And also the physiological trait phenotyping is also conducted. Then we make the genomic selection model. Oh, I forget that still Japanese data is remained. Then from the training population, we create progeny, second generation progeny. And then at the same time, we also the genotyping for the second generation progeny. Then we apply this genomic prediction model. And then finally, we can conduct the selection using the genomic data. This is our workflow in this project. Thank you very much for your attention.
Thank you, Professor Naokitani. The next is Dr. Shinichi Kakuta from Sumitomo Forestry Corp.
Thank you. Selamat malam. Is it correct now? Okay. Oh, okay. So, okay. I start presentation. Firstly, I would like to explain about our company. We originally started in 1691 with the management of the forest affected by copper mining in Mount Beshi in Japan. The photo on the left is the Beshi copper mine in the 1881. As you can see, excessive harvesting, smoke and other pollution from the copper refinery devastated the surrounding forest. Having been that situation, our second general manager said, while we should continue to reap the benefits of our country's blessings, our destructiveness is betrayal unto God. He executed a great reforestation plan in 1894. We used available technology to restore the greenery on the devastated mountains to protect the natural environment. Since then, it remains a part of our basic philosophy to grow and make use of tree in order to contribute to society by improving the environment. The photo on the right is the current state. Next, I will explain our business. Our enterprising does operate from tree plantation to making final product such as timber, engineering wood, and wooden furniture products. Cooperation of each sector is a strong point to making good products with a timely and cost competitive manner. And this shows our log and lumber mills in the Asia Oceanian. Our long histories in the modernized techniques also help manage forest and plantations in Asia Oceania countries in sustainable and environment-friendly manner. As a result, we have FSC certified forest in all of our operation countries. One of our affiliate company is PT Kutai Timber Indonesia in Probolinggo. KTI manages approximately 6,500 hectares of plantation area, in which several fast-growing species, including Sengong falcataria, is planted and controlled under FSC certification. At the stage of implementation of this project, we plan to have KTI cooperate as a receiver for implementation. Next, I would like to explain our role in this project. Our aim is to establish mass production system for excellent varieties of seedlings selected by genome selection breeding. Specifically, we will develop vegetative propagation methods such as cuttings and tissue culture by using embryos, apical buds, and somatic cells to produce clonal seedlings of genetically superior, fast-growing trees. The reason for working on these technologies is that some tropical tree species are difficult to grow on cuttings, which makes mass production of chrome difficult. Although this approach may be a little bit very high, hurdle is very difficult. If mass production can be realized, this result is expected to be suitable not only for reducing production cost and time, but also for maintaining good genetic resources over a long period of time during genome selection. This is an example of coffee. In this case, mass production of zoomatic embryos of elite clones can lead to the reduction of production cost of clonal seedlings into a reasonable cost. For example, as an initiative in Japan, we are contributing to the conservation of cherry blossoms and plum blossoms, which are famous for Japanese shrine and temple. Thank you for your attention.
Thank you, Dr. Shinichi Kakotan. The next is Professor Yossi Kosumura.
From University of Tsukuba. Hello everyone. My name is Yossi Kosumura from the University of Tsukuba. And today I talk about the genetic diversity and the adaptation across genetic resources. Today I have only just 10 minutes. I just briefly and talk about this our project. And firstly, the biodiversity consists of three diversity. First, ecosystem diversity. You can see the many kinds of ecosystems, such as a forest ecosystem, marine ecosystem, human-related ecosystem. And we can identify that kind of ecosystem. And also, we can identify the species diversity. We can identify the tree species, also the insect, animal. But and the genetic diversity, this is the we cannot see. Mostly the genetic diversity we cannot see because it's just the difference in nucleotide. Therefore, the very few genetic diversity we can see. For example, such as the hair color of animal or our human, that's controlled by single gene. Only in that case we can identify the genetic diversity. But in the other case, mostly, we cannot identify that genetic diversity. But this genetic diversity is very important, fundamental factor for the diversity. Therefore, we have to understand the genetic diversity for our target species. I show you some example of genetic diversity for genetic differentiation between population. This is the case of the Fagus coronata. Then we collected some many samples to cover the natural distribution area. So we collected study material from all many populations. Then we investigated genetic diversity. Then we found genetic diversity is higher in the western part of Japan. Then gradually decreased to the north, just like that. Then the mitochondrial DNA, also the western part of Japan, they have the many, we can detect many haplotypes. But if you go to the north, there's a haplotype only just one. Very simple. That means that this species is very clear and they have the very clear geographic line of the genetic diversity. Then another example of the Cryptomeria conica, which is the most important forestry conifer in Japan. Then we also did this species and we collected many material from the whole distribution area. Then we found there's 4 genetics groups. This is the blue one. This is the most northern population in the Pacific, Japan seaside. They have the blue type, but the other Japan seaside they have the green. Then Pacific Ocean side, mostly population just like a red. Then another important one, the Yakushima population. This is the southernmost population. They have the yellow type of genetic variation. Then how about the genetic diversity? Genetic diversity also the higher in the western part of Japan, and especially especially the refugee area in the western part of Japan during the last glacial period. Last glacial period means that 20,000 years ago. And in this population, for example, Ashu population in the peninsula, Okinoshima Island, and Yakushima Island have high genetic diversity in terms of the rare allele frequency. But the other population very low genetic diversity. That means this species also the very clear geographical line. Then how about the monological trait? Then in case of the Fagus grenata, northern part of the population, they have the large size of the leaf. But if you go to the southern part of Japan, this leaf size is very small. They have very clear geographical line in this case. Then this is a Cryptomeria conica. There's the 2 types of phenotypes. That's one is the Urasuri group. This is distributed along the Japan seaside. Another one, the Omotosuki group. This is the distributed Pacific Ocean side. Then Omotosuki variety. This is the needle is very hard and branches very large. But the Japan seaside, also needle is very soft. Just branches like just gentle. This is the result of the natural selection, because winter season, Japan season has a lot of snowfall, but the Pacific Ocean very little snow. That's the maybe reason of this difference of the phenotype. How about the Southeast Asia, like Indonesia? Then we studied some Dipterocarp species. Then we found, and in Shoria Repulsa, there are 2 genetic groups. One is the Borneo type, another is the Peninsula Malaysia, Sumatra, and Java. This is a result of the chloroplast DNA. But nuclear DNA also very similar result of the chloroplast. Then another example of Shoria macrophylla. Then we collected materials from North Kalimantan, Central Kalimantan, and West Kalimantan. Then North Kalimantan have a very specific and special genetic structure compared with others. We can identify 2 genetic groups in this case. Then mostly 3. They have the very clear genetic structure. Therefore, we should conserve such kind of genetic structure in the future. But if we move some material to the very different environment, probably that genetic disturbance may occur because of the genetic constitution. Native population may change by the invasion of non-native individual. Adaptive loss in the native population may disturb by this invasion. Then finally, outbreaking depression may occur in this disturbance. This is one of the examples of the outbreaking depression. I don't talk so much detail in this time because I don't have enough time. But if we planted a non-native material to some local population, F2 generation some individual cannot adapt into such local environment. Then finally, genetic diversity gradually decrease. Then, and that population decline. That's a big problem in the future. I show you the one example of the planting experiment of a beech tree. Then this is actually one of the scientists, Koyama, he done this experiment. He collected material 2 points. One green one, sampling point A. This is a very little snow area. Then another one is the sampling site B. This is a very heavy snow area. Then he, using this two kinds of the material, he established a 3 planting sites. The Sakai site, and the Shiojiri site, and the Koumi site. Then when we look at the Sakai site, this is the A group, A material from the little snow area. Most of the seedling damaged. Maybe they cannot survive, or if they can survive, but a very bad growth. But another one, material B, this is a very similar environment from the heavy snow area. Then more than 6% showed very good growth. Then another planting site, Shiojiri, then same environment from the sampling site A. This seedling is quite good, but the material from the sampling site B, more than 70% seedling was damaged by the snow. This means if we move some material to very different environment, they cannot get a good growth. Therefore, we need a genetic guideline for the seed and seedling transfer, not only our temperate country, but also the tropical area. Because the plant species is adapted to each local environment as a result of the natural selection. Generally, each local population is genetically differentiated between regions as a result of the historical events, such as climate change. Then such important genetic differentiation should be conserved for the human future human resources. Then in Japan, I published this book for the genetic guideline for 43 species for the forest conservation in 2015. Therefore, we try to do the similar study in this area. Then this project we have the 2 groups. One is the modeling group to predict the future planting site. Then second group is our genetic group site. This is the image of our Satreps project. Actually, this picture made by the Dr. Tani. Then our target species is this one, Tectona grandis and Parcataria and Shura reposora. This is the first priority species. Then if we have enough time, we continue to this study to the another 3 species, Shura macrophylla, Prados, and Cassia. Thank you very much.
Thank you, Professor Yossi Kosumura. The next is Dr. Toshiaki Kondo from JIRCAS.
Okay, very good afternoon, everyone. Thank you very much for your joining to this seminars. I'm Toshiaki Kondo, belongs to Biological Resources and Post-Harvest Division of JIRCAS. So first of all, I would like to express my appreciation to Professor Budiadi, Professor Widi, and many staffs of UGM for taking time to organize this ceremony and this seminar. Terima kasih banyak lah. So Anyway, today I'm going to introduce about my research proposal for sub-project 3, entitled Evaluation of the Impact of Forest Restoration on Economy, Society, and Environment. So the main aims of our research group is to create incentives for forest restoration utilizing native tree species which hold higher climate change adaptation abilities by evaluating the impact of forest restoration on economy, society, and environment. So to achieve this purpose, we established 2 research groups. One is the evaluation of environment. Another one is the evaluation of economy and societies. The member is shown in here. So unfortunately today, researcher from research group 2 cannot come to Indonesia. So today I'm going to introduce about the first group research proposal. So as you know, tropical rainforest in Southeast Asia with high levels of biodiversity provide many types of ecosystem services. So it's quite difficult to conduct a research on all species and all types of ecosystem services. So in our research proposals, so we will focus on the soil microbes and subsequent ecosystem services characterized by the GHG emission and uptake via soils. So firstly, I want to introduce about the characteristics of soil microbes. The carbon stored below ground is transferred to the atmosphere by microbial decomposition of soil organic carbon. This phenomenon is caused by heterotrophic respiration, and it is far more than that released by annual fossil fuel CO2 emissions. Furthermore, some soil microbial species, such like methanogen and methanotroph, are strongly related to GHG emission and absorptions. So I think so forest restoration would bring about large changes in soil microbial community, and subsequently their ecosystem function related to GHG flux would be improved, I think. So traditionally, such dynamics of microbial community is always analyzed by using a micrological cultures. But however, because of the limited variety of culture media, less than 10% of all species within soil samples can be detected by using this method. So it means that it is quite difficult to monitor soil microbial dynamics by using this method. But by using a recent developed genetic analysis, so for example, so by using a quantitative PCR method, we can easily detect the amount of soil microbes at individual levels. And also by using metagenome techniques, we can identify the species of 20 to 200,000 individuals per soil samples. And also, based on this database, we can easily predict the function of soil microbial communities, such like decomposition, methane production, and nitrification. So anyway, so I want to show some examples here. So this figure shows the comparison of soil microbe composition among 4 different land use types in Malaysia. One is a primary forest. Sorry, I will prepare. One is a primary forest. Second one is a secondary forest. Third one is a rubber plantation. I also will collect the samples from oil palm plantations. By using metagenome analysis, we can easily detect the changes of soil composition, soil microbe compositions at species level. So we can easily detect the changes according to the land development. And also by using a database of metagenomes, we can easily identify predict the function of microbial society, such like CO2 emissions, methane emission, methane absorption, and N2O emissions. Of course, in this Satreps project, by using such chamber systems, we are going to monitor CO2, CH4, and NO flux directly. Anyway, by using these two methods, we are going to evaluate the forest restoration. Okay, so finally, I want to summarize my research. So in the research group 3A, we are going to evaluate the impact of forest restoration utilizing native trees on biodiversity and related ecosystem function. So especially we are going to focusing on the advancement of soil microbial community function after restorations by using metagenome analysis and portable chamber systems. So fortunately, we already developed these portable chamber systems in Japan, which can monitor CO2 emission, CH4 absorption, and NO emissions simultaneously by using one small chamber. Anyway, so maybe next month or next December, I'll come back to here with this chamber, and I will try to monitor the GH flux in some restoration site. Terima kasih banyak lah. Thank you lah.
Thank you very much to all of the experts, and we would like to extend our gratitude to His Excellency Professor Naoki Tani, Dr. Shinichi Kakuta, Professor Yossi Kosumura, Dr. Toshiaki Kondo, Professor Dr. Muhammad Naim, and Dr. Widyatno for gracing this occasion. Arigatou gozaimasu. And next we will have a group photo session. We invite all of the experts and also Dr. Sabto Indriyoko, Dr. Budiadi, and all of the participants. Thank you. Thank you for your attention and good afternoon. Wassalamualaikum warahmatullahi wabarakatuh.